welcome May-Britt Moser.
Ni hao. Dear, respected audience, I feel so honored to be here today, and I'm so grateful for all these events that you have created here. And this dance that you made for me almost got my tears to my eyes. It was so beautiful. China and Chinese people are gorgeous. So I'm so happy that I'm allowed to tell about the tipping points in my career. What was important for me? And there's one thing that we all share inside our skull, and that is the brain. And it's such a wonderful organ. Without it, we are nothing. So that is why I'm so curious of how can the brain do all what it's doing? And hopefully, when you leave the room today, you and I can agree that, okay, we understand a tiny bit more what the brain is doing, the mysteries in the brain. So I would like to say that every science starts with a child and the child's curiosity and the passion for questions. And the same thing happened with me. And as a scientist, I can follow my curiosity and passion in my job, which is a blessing. One tipping point in my career was that I met May-Britt Moser, and we decided to work together. And we were accepted by our supervisor, the medical professor Per Andersen in Oslo. And he said that you are a psychologist, you know behavior. He's a medical doctor, he knows the brain. And he was, of course, a famous neuroscientist. And if we work together, maybe there will be something really nice happening. Our vision at our institute, and it has in fact been the vision since I was a child, that we try to understand the recipes, the algorithms that the brain is using to create higher cognitive functions. And where should we start when we want to learn about the brain? My start was in the hippocampus, and I think that was a good start. Why is this structure, brain structure, so important for us? It's just behind our ears. We have two of them, and it's the most beautiful structure in the brain. It looks like a seahorse, and that is why it's called a hippocampus. In the 50s, there was a patient, or many patients, but there's one very, very well-known patient called HM, and he had epileptic seizures that required an experimental surgery, and the surgeon removed the hippocampi on both sides of HM's brain. His intellectual function was much better because he was not disturbed by the seizures. But whenever he met the doctor and the nurses after the surgery, he couldn't remember that he had seen them. He couldn't find his way from his room at the hospital to the restroom and back. So every new experience for him was like a new thing, even though he had just seen a person, even though he had just been to the restroom. So for him, losing the hippocampus on both sides left him with no ability to create new memories of events, and he had no sense of time, what is coming before something else, and space. So this information puzzled neuroscientists for many, many years. Then John O'Keefe, whom we received the Nobel Prize with in 2014, he decided that he really wanted to understand what is this hippocampus doing, and how could he address that question. He decided to put small sensors close to the nerve cells in the hippocampus, and then he wanted to observe the animal when the animal was walking around in a normal box chasing chocolate or rice that he used. And then he was listening to one single electrical potentials. And the first talk today, we heard about electricity. Our brain is also talking, the cells in our brain, they're talking to each other through electricity and chemicals. So we can listen to the activity of our brain like we do now. Here you see a rat that is implanted with such small sensors, and one red dot, that is this activity of this one hippocampal cell. And this terrible sound is music in my ears because it tells me this is where this cell is active. And what you see when this animal has been walking around for a while is that there's a space where this cell has been very active, and there's some space in the box where the cell is silent. So if we had observed only one such hippocampus cell, we couldn't say much what was this cell signaling or responding to. So John, he heard many such cells. He recorded from many such cells in different animals. And he saw that each cell would have its own preference for different places in the environment. That means that these cells code for the location where the animal is. And we know that humans, I and you, we all have such cells in our brain. And before I move on to the next slide, I have to describe another way to show this activity. So if you see this middle panel, then you see these white traces. That is the trace or the path of the animal, because we have a camera above that is getting the XY coordinates of the animal. And the panel to the right, that is a color map just showing that warm colors, that is where the cell is most active. So we knew about John and his discovery, and he was a good friend of our supervisor, Per Andersen. And Per said, because we really wanted to learn this technique, he said, go to the best one. And of course, John was the best one. And through his network, he allowed Edward and me to come to London. And we brought our 2 children with us to London. And John personally trained Edward and me to do such recordings that he had done himself. And after this visit to John's lab, We got 2 positions in Trondheim, in Norway, at the University Norwegian University of Technology and Science. And we brought with us this question: where is this spatial information that we see in the hippocampus, where it's coming from? How is it generated? Through many control experiments that I can't tell you about because we don't have the time, then we decided that we should record in another brain structure that is called the medial entorhinal cortex. Why was that interesting? Because this is the cortical input to hippocampus. People had been there before, and they said there's not so much interesting there, but we wanted to see ourselves. What happened when we recorded in this structure was magic. You remember the place cell that John discovered, the field in just one tiny corner of the box? The cells in entorhinal cortex, they were different because they had many fields. What was so strange and remarkable about the firing activity of these cells in entorhinal cortex is that they had the many fields, they were so regular. So this cell was tessellating the environment. So you could use this as a pattern in your bathroom, and it would be beautiful. And you see that we can fit in equilateral triangles into this pattern because it's so precise. And this is biology. This is not a machine. This is not physics. It's biology. And still, it's so precise. We were so shocked when we discovered these cells, and we decided to call them grid cells, the coordinate system of the brain, and they can be used for the metric when we move around, which is very useful for the hippocampus when it's creating the place fields. So I learned yesterday that I have to be a bit patient. I would like to go to the next slide, but yes. I would like to tell you about other cells in the brain and also, finally, also in the entorhinal cortex, which are important for this GPS system that we think are in the brain. The head direction cells, they are one of my favorites when we get journalists in the lab. If you imagine that this is the rodent, a mouse or a rat that I have here, we record cells here in the entorhinal cortex of this animal, and then we pick out one cell. This cell has a bias to be active when the head of the rat or mouse is in that direction. So then it's like this, pop, 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 pop. And then we turn the animal, silence. Pop, 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 pop. Nothing. And there are different cells being active to different directions. For all directions, we have head direction cells. And these were discovered in New York by Taube and Rank in the 1990s. And we also found them in the entorhinal cortex. In a bit, I can tell you why this finding was so important for the grid cells. But before I do that, I would like to remind you of an animal walking around, even a human, in an environment. You can stop up, you can look around, you can eat something, greet somebody. So the speed is changing all the time. And how is it then possible for these grid cells to be so precise that we can fit in these equilateral triangles when you have this change in speed? There must be a speed signal in the brain that is feeding into these grid cells. And how can we study speed in an animal? We have to control the speed of this animal. And if you have ever worked with rats or mice, it's very difficult to control them, to tell a mouse, you have to run fast now, or you have to run slow. So what can you do if you can't tell them to do it? You put them in a car. And we need a Flintstone car. I don't know if you know a Flintstone, but that is a car where Flintstone was running in the car. Why do we need a Flintstone car? Because otherwise the animal falls asleep. And then if it's sleeping, then we don't get the effect we want. So then we put the animal in this frame, in this car, the animal is running, and we control the speed by pushing this car at different speeds by the computer. And in this way, we discovered cells in the entorhinal cortex that would increase their activity when the animal is moving faster. And that you see here to the right, this figure, that when you have the speed of the rat on the x-axis, you have the activity of the cells on the y-axis, and you see there is a linear function. And of course, rats, they don't drive a car in normal life, so we would have to check whether this is true also when they walk around spontaneously, and it's true. There are other types of cells, so it's like a jungle of different functional cells in this structure. The border cells, why do we need border cells? Do you think I need a border cell? I think I need. So yesterday I heard, don't pass this line. Then you fall down. And now I have border cells in my head. These cells, they will signal the boundary of your environment. And on the screen, you see one such example of such a cell that has a bias towards the east wall in such an environment. And different cells have different biases and signal different parts of the environment. And there are cells that tell you the distance and the direction towards an object. So if there would be an object here, then one cell, if we record, would then be active when I would be at a certain distance and also in a different, or in the same direction. And it doesn't matter if I'm turning around, this cell will still signal the distance and the direction. If you move the object, the cell will still prefer the same direction and distance. And again, different cells with different directions and different distances. So over 50 years of exploration in this brain structure, in the hippocampal formation, we have then discovered all these kinds of cells. What we also know is that when people get dementia, Alzheimer's disease, these cells are the ones that die first. And when you hear about people getting Alzheimer's disease, the first thing that happens is that they lose their sense of time, they can't find their way, even in a familiar environment, and they have problems to create new memories. So when we search for the causes of dementia, we have to go to entorhinal cortex and ask the question, why do these cells die? And that is when we can stop them from dying. So this is ongoing research. This is 2014. So, Chichi. So we were awarded the Nobel Prize together with John O'Keefe because of our discoveries of these cells that I have described to you. And we had a lovely time in Sweden, and here you see the royal family that we were also allowed to visit. So if our determination to work in this field was because we wanted a Nobel Prize, we could just go home and enjoy life. This was it. Of course, it was not. As I told you, we scientists are like children. We are still curious. We still have the passion. It's almost like being insane. You can't stop asking questions and getting answers. So what type of questions do we now work on? What I didn't tell you is that in our brain we have more than 80 billion nerve cells. These cells need to talk to each other. What I have described to you now as being single functional cells, but what is the most interesting part is how these cells talk to each other to generate our cognitive functions. But then the difficulty we have is then we need new tools. We need new technology because we've been able to only record single cells earlier. Then we have to go to China. Here you see a researcher in Trondheim who got his PhD at the Peking University, supervised by He-Ping Cheng, and we call him Peace. And Wei Jian, in his PhD, he was, together with his supervisor and his group, they were able to reduce a 500-kilo two-photon microscope down to less than 3 grams, in fact 2.6 grams. And I brought one such microscope with me that Wei Jian made. And you see, it's tiny. And why is it important that it's tiny? If we want to study spatial navigation and the GPS system, the animal has to explore freely. Otherwise, we can't do it. We can't hook an animal up in a big microscope. We have to let the animal run around. And Wei Jian, he implanted a small prism in the brain, and he could then put this microscope on top. But when he came to Norway, and we had to collaborate because the microscope that he had developed here together with his people at Peking University wasn't still ready to be used in animals. And this Wei Jian was able to do in Trondheim in Norway together with us. And what he also did in our lab together with our collaborators, the researchers in our lab, was to use mutant mice, either with viruses or genetically manipulated. And you saw the dancers, they had small lamps. And in the brain of these animals, it is like they have small lamps. When the cell is active, it's blinking. And with this device that Wei Jian had, the Mini-2P, then we can study this. And Wei Jian was able to record hundreds of such grid cells that I just described to you. And now we can ask questions. And you know, John Hopfield, he got the Nobel Prize this autumn, and he suggested how different networks work together in order to make functions in artificial intelligence. This is also relevant for us. And here we have got theories on how this should function. We can also use this microscope to see where the cells are located, different functions, where are the head direction cells, where are the grid cells, and how they talk to each other. So that is a project that we are still working on, so I can't tell too much about it because we are not allowed to, but maybe later. There is also another technique that we have developed in our lab because we were allowed to be part of a consortium led by Janelia Farm. There's a company in Belgium developing small microchips that you can plant in the brain. And why is that important? Because then we get a time resolution so that we can see these tiny electrical potentials in many thousand cells at the same time. And then we can ask, how do these work together? And they are correlated, so we can use low-dimensional technology or analysis, and boom, you know what? We got a torus in these analyses when we analyzed the activity of these cells. So if you see this beautiful torus here, it's made by UMAP, it's a low-dimensional method. Each dot here is a time point when these cells are active, and then the activity is going back. So there's a manifold deciding when the cell can be active. And you see that the path in the environment follows the path on the torus. I see I have to speed up. What is so interesting is that this torus, this GPS system, is on also when we sleep. And why is that so interesting? This slide is showing my grandchildren. And the reason why I show you the grandchildren is that because we have now discovered this torus, this manifold in the GPS system, we can start to address questions that philosophers have been asking for thousands of years, because they ask, are we born with something so that we can experience the world, or are we tabula rasa, a white sheet? And then Immanuel Kant suggested, no, we are born with the ability to experience space and time. And that is exactly the questions that we can now study, because we can study animals that are so tiny, so babies, that they can't even move, they just sleep. And then we can ask, do we see the torus in these animals? This is, again, in progress. We are not allowed to tell you too much about this because it's streamed, but it's so exciting, I can promise you. So I would just like to say thank you, and I would like you to leave this room with two things, two messages, before I say there's a slogan in our lab which is telling that we should have excellent science, happy people, happy animals, and diversity. We have curiosity, great questions, passion, talents, and technology. We need to collaborate. But there's two things. If you remember the structure hippocampus, then your brain is intact. And the second thing, I want you to leave this room being so grateful for this fantastic organ that we all carry around. So thank you so much for your attention.